So I mentioned to you this other end of the cell. The other end of the cell is, we call it a synapse. And that's when one nerve talks to another. Okay? And so the same technique applies here. We're using staining to say, are these cells talking to each other? And we can do that by staining a bunch of proteins that are present at synapses. And it turns out that every cell, every synapse marker that we've looked at that is at this complex between photoreceptors and the next neuron down the line are expressed by these transplanted stem cells. And another pretty picture here just showing all of these red cells out here are new cells that have been transplanted in. And all the green staining you have here are connections that those cells are making with the host retina. And I'm going to trip over this eventually. And just another image. This is what we have to do when we publish this work. We have to show the same result about 7 or 8 different ways to convince reviewers that we're really proving what we claim we're proving. And so this is another study. Now there are also a couple of antibodies that are used called Piccolo and Bassoon. I'm not quite sure where these names came from, but this is Bassoon antibody in blue. And again, what we're showing is the cells that we're transplanting are making a connection, they're making the right kind of synapse, and they're making it on the right cell type. So we have no way of knowing a priori whether these cells are going to be able to find their targets once we transplant them. Maybe they're going to connect to the wrong cells. Luckily for us, and it is luck, nothing we've done, the cells know what cells to connect with. Okay, and that's a hallmark, I would say, of central nervous system repair, is that the brain and the central nervous system will do everything possible to extract information from a new input. And that's really quite remarkable. And I think the gene therapy study that was done in monkeys that rescued color blindness from these guys really proves that point. Because it turns out these animals don't get new circuitry. They don't get new neurons. Somehow the brain that's evolved with 2 types of cones gets 3 types of cones and can make use of it within a matter of weeks, it turns out, can figure out how to use this input. So I think from a point of view of repairing the brain, repairing the eye, there's a tremendous amount of hope in the field that, first of all, whatever we can do is going to be helpful. In terms of a minimal amount of function can be very helpful, but also that the upside, the possibility of the brain actually extracting more information is really there and has been proven by a number of animal studies so far.